We live in a world full of bacteria. Bacteria are everywhere, around us, on us, and inside us. Although some bacteria cause infections, most species are harmless or perform functions that are beneficial to our body. One of the most important functions of beneficial bacteria is to initiate, teach, and strengthen our immune system. For example, some studies have shown that mice raised in a sterile, germ-free environment have very poor immune system. The microbal community, microbiome, that lives on us and in us play a fundamental role in stimulating and training the host's immune system, as well as ensuring the balanced functioning of the immune system. In return, the immune system has largely evolved as a means to maintain the symbiotic relationship of the host with this highly diverse and evolving microbe. In the modern world, the way we live has changed significantly since the last couple of decades, and it does not remind anymore the lifestyle of our ancestors. Recent research has shown that people living in high-income countries continuously strive to avoid and prevent bacteria occurrence in our environment, and thus also eliminate contact with beneficial bacteria. The modern human living in the city has developed a microbiome that lacks the resilience and diversity that are necessary to establish a balanced immune response in our bodies. Active bacteria avoidance, constant cleaning, washing, and disinfection is proposed to account for some of the dramatic rise in autoimmune and inflammatory diseases in urban inhabitants, where our symbiotic relationship with microbes has been the most affected. Let's see what kind of changes in our way of life affect our microbiota. Let's start from the very beginning, from the birth. It has been observed that the birth rate by cesarean section is increasing around the world. Some scientists have studied the long-term effects on babies born by cesarean delivery compared to babies born naturally. It was found that the babies born by cesarean section had an increased risk of developing asthma before the age of 12, an increased risk of obesity before the age of 5. Available data indicate that atopic disease is more common in infants after cesarean section. In addition, babies born by cesarean section were also found to have significantly high rates of asthma and allergic rhinitis, celiac disease, and gastroenteritis. The reason for this phenomenon, according to the researchers, is that the babies born by cesarean section do not receive the initial bacterial contamination, since they bypass the main source of beneficial bacteria. The infants born in a natural way pass through the channel filled with diverse and rich bacterial community that plays a decisive role in the development of the immune system of the child. These natural bacteria colonize the newborn and form its immune system. It was found that the babies born naturally were colonized mainly with bacteria Lactobacillus, while babies born after cesarean section were colonized with a mixture of potentially pathogenic bacteria commonly found on the skin and in surfaces in hospitals, such as Staphylococcus and Acinetobacter. This initial difference in infants' bacterial contamination plays a crucial role in the further development and functioning of the immune system. As urbanization increases, more and more people are spending their time indoors. It has been found that modern buildings and modern lifestyle contribute to the development of microbiota that are harmful to us. In one study, researchers examined the presence of antibiotics-resistant genes in the dust from a floor in a Norwegian kindergarten. Antibiotic-resistant bacteria have been found in samples from most of the rooms, even though the kindergarten had only been opened. The results of this study indicate the presence of antibiotic-resistant bacteria such as methicillin-resistant Staphylococcus aureus. 3 different types of multi-resistant bacteria have been found in the dust. These results suggest that floor dust from buildings may serve as a reservoir for the spread of multi-resistant bacteria. Another study analyzed the dust samples from the intake and exhaust air filters in 6 Norwegian nurseries. About 96% of bacteria found in the dust were classified as actinobacteria, proteobacteria, Yersinia, Helicobacter, Legionella and many others, Firmicutes, Bacteroidetes. About 30% of the bacteria in the exhaust filter have been classified as the bacteria of human origin, such as Streptococcus and Corynebacterium species. They were practically absent in the outside air samples. The results of these studies showed that humans themselves shape the microflora in the indoor environment, creating the environment relatively rich in bacteria of human origin with high health significance and not necessarily positive. Scientists have also started to investigate how the increasing energy efficiency requirements for the buildings affect indoor air quality. To reduce energy consumption, modern houses are very well isolated, and air is supplied to the premises through mechanical ventilation and air recovery system. Due to efficient ventilation system and air filters, there is created a barrier between the outside air and the indoor air. Air filters that clean the incoming air also prevent outside bacteria from entering the room together with the fresh air. It has been observed that there are very few naturally occurring bacteria and particles indoors in modern buildings. Instead, human pathogens invade and thrive on surfaces in homes and ventilation systems in our buildings. Several studies that analyzed microflora in our homes found out, for example, the presence of multi-resistance bacteria in ventilation systems and on some indoor surfaces. The microbal community in our homes and other buildings is no longer influenced by nature, but mainly sourced from humans. A study that analyzed British homes showed that the microbiota in each home was different, and it was defined by the family living there. Analysis of the bacteria has shown that the humans are the main source of the bacteria. It was also noticed that when the family moved, they took their bacteria with them and planted them in a new home. Public transportation in the cities is also a source of human bacteria. There have been many studies analyzing bacterial contamination in various types of transport around the world. Major finding is that hand-touch surfaces in public transport usually contaminated with staphylococci and enterococci bacteria and can be reservoir for community-associated multi-resistance bacteria. As it was noticed, average American and European urban inhabitant spend about 90% of their life indoors. This means that we are mainly exposed to microbiota of human origin, and it looks like that we live in a kind of bubble of human pathogen bacteria with little or no real contact with nature and beneficial bacteria. Children living in modern cities, playing in the streets, no longer have natural contact with the nature. Modern playgrounds are designed and shaped to protect children from an injury, but at the same time, they limit their natural exposure to dirt, which is a source of beneficial natural bacteria. Research shows that the dirty children are healthy children. This is because the dirt is the source of beneficial bacteria for us. The germs found in the mud play a crucial role in teaching the children's immune system to respond correctly and to populate the good microflora in their gut. Studies have shown that kids who grow up on a farm or who have the dog at home are 20% less likely to develop asthma. Another important factor affecting our health is the food we consume. In the grocery store, basically almost all food is pasteurized, packaged in a protective atmosphere that kills virtually any beneficial bacteria, so our nutrition does not contain the required amount of beneficial bacteria anymore as in old good times. There are many studies investigating impact of sterile food intake to our health. This is a huge topic beyond the scope of this video, but I hope I can make a video on this later. The modern, ultra-disinfected world protects us from the exposure of beneficial bacteria. Researchers have found that a few specific autoimmune diseases such as asthma, hay fever, inflammatory bowel disease, and various allergies have become much more common as we become more sanitary, striving to maintain ideal cleanliness and hygiene. So what to do? There are many things you can do to build a balanced microbial community around you. First of all, be more outside, in nature, in the park, take daily walks, play more often with the children outside. Don't be afraid to get dirty. If possible, go to the countryside or visit a petting zoo where you can clap animals. Do not try to maintain a sterile home. After all, along with harmful bacteria, you are also destroying beneficial bacteria when you disinfect your home. Try to ventilate the room more often so that the bacteria we need come into the room together with the fresh air. Avoid using antibacterial soaps or shampoos or other hygiene products that kill bacteria. If you can, buy fresh vegetables and fruits from the market, from trusted sources. Consume more fresh foods as well as fermented foods which contain a lot of beneficial bacteria for us. I hope you have learned something new today. Please tell me what other topics would be interesting for you and I'll make video for you. Thank you. Bye.